Hello, this is Tommy. Welcome back to Chat-Omics. In today's video, we will continue our journey replicating a published genomics figure using ChIP-Seq data. So in previous videos, I have shown you how to download the FASTQ files to quality control using FastQC, and how to align it to the genome using Bowtie 2, and that creates a SAM file, which is an alignment file. It's a just regular text file, tells you for each read where it aligns to the genome. And then you can convert that SAM file to a binary format called BAM file, and then you sorted the BAM file, then you get this sorted BAM file, okay? Once you have the sorted BAM files, you can do two things for ChIP-Seq data. So the first thing is to generate the BigWig file, and second thing is to call peaks. So I will show you what are those two things. So first, what is a BigWig file? Okay, so a BigWig file is just a binary format that is commonly used in the bioinformatics to efficiently store and visualize the continuous data over genomic coordinates, such as read coverage or signal intensity. In this case, where the chip signal is enriched in the genome. So it's derived from this regular text file called the Wiggle format, but optimized for faster access and reduce the file size. So you can actually read the link here to understand exactly the format. You know, bioinformatics is notorious about different file formats, but you can read the other link here to understand the various formats. For example, the BigWig is one data format, and then after we use actually max3 or max2 to call peaks to give you a BAT file, it's a different bioinformatics format. Okay, to create a BigWig file, we're going to use this tool called DeepTools. It's a Python package and is very versatile and has many subcommands. And we will use this subcommand called bank coverage to generate BigWig files. But I want you to kind of understand how exactly that BigWig file is actually calculated or generated. So this is just to install the DeepTools using Conda. You can just follow the commands here. But let's talk about what is a BigWig file and how it is calculated. Okay. So remember, after we align the reads into the genome, we get a SAM file, and the binary format is the BAM file. And essentially, those files, they contain the reads information. So where are those reads mapped to in the genome? So it will tell you the coordinates that this read is mapped to, right? So the BigWig file essentially is just trying to reduce this information from the BAM file to the simple information. How many reads do I have per bin or per genomics bin here? So in this case, for example, in chromosome 2 here, in this 20 base pair bin, you have 5 reads, for example, in that bin. So you have a number 5 here. And in the next 20 base pair, like you have a signal 3.2, for example. And this is because you have digits, because you do some sorts of normalization, so you'll get those digits. So I will talk about the normalization methods actually in a minute. Okay, so essentially that's the idea. So for example, in here, you have 50 base pair per bin in the genome, and it just counts how many reads fall into that bin, and then you get this more like a histogram type of thing across the genome. So you can get a sense of the signal across the genome. Okay, so how is that computed? For example, in ChIP-Seq, usually you actually sonicate, essentially break the DNA fragment into 200 base pair fragments, but we only sequenced the first 30 to 50 base pair reads, like in the early days. And also, it could be also single ends or paired ends. In our case, actually, it was single end. So in this example, say you have actually many bins here, okay? And each bin is 10 base pair, 50 base pair here. And now, for example, now in this example, you have actually a paired end read. So this is one pair, and this is one pair, this one pair. And that paired end reads can span actually multiple actual bins, see here. So for example, this first bin here, the second bin here. But again, it's actually straightforward. You just stack those reads together and just count how many number of reads in each bin. So for example, in the first bin here, you only have 1 reads here. So the number is 1. And then in the second bin here, you have 1 and 2, so it becomes 2. And for this one, for example, you have like 1, 2, 3, 4, 5. Then you have like five reads, so that the number is 5. The same for here is 1, 2, 3, 4. 1, 2, 3, 4, 5. I think this is probably also calculated. It's right in the edge here. So you just do that genome-wide, okay? So after you get this number, and you can somehow normalize it because different experiments, they have different total number of reads. For example, you want to actually normalize that. So there are different ways to normalize it. So just two methods here. So 1x coverage, or RPKM, reads per million, reads per genomic, reads per million, per kilobase per million reads, okay? So the formula is actually, okay, the number of reads, for example, per bin, divided by the number of mapped reads, this total number of reads in the library, times bin length. So the bin length is actually in kilobase pair. So if it's 50 base pair, it will be 50 divided by 1,000, okay? Or you can use this RPGC methods, is reads per genomics content, okay, to 1x depth of the coverage sequencing depth. So this is actually just the total number of map reads, again, to the library size, the total number of reads, and then times the fragment length. Okay, for example, if it's 200 base pair, times 200 base pair, and divided by the effective genome size, how many total base pairs in the genome. For example, human genome will be 3 billion base pairs, okay? So just different ways to normalize the sequencing depths, okay? And now that you have understand how the BigWig file is generated, you can use this command, band coverage, to calculate the BigWig files. So the command will be band coverage, then give the band files, and then you can use normalizing using different method. In this case, I would use RPKM, okay, reads per kilobase per million, okay, and then extend the reads into 200 base pair. So because in our experiment, it's actually single end, so only the, I think, 30 to 50 base pair is sequenced. We want to extend that to the real DNA size that we sonicated 200 base pair. And this output will be a BigWig file, okay? So, okay, if you go to command line here, band coverage, so if you just type band coverage after you install DeepTools, it will actually, so you can just do help, it will tell you all sorts of different parameters that you can change, okay? But in our case, you just need to use this. And because we have multiple band files here, again, this is a very commonly used Unix trick, is just use a for loop. So you can use 4 band files in all the sorted band files and do this command. And again, we're using the batch stream substitution here. So substitute the sorted band to BigWig, BW. And so actually we can take a look at the commands. So I will just do echo. Actually, it will just print out the commands, will not run the command. So if you do this, essentially this for loop will give you actually the commands, okay? Okay, that's how you actually avoid the typing all those commands manually, okay? If you have like 10 samples and you don't want to type this 10 commands, rather than use this for loop. So if you look at what are the sorted band files here, LS here, then we have 1, 2, 3, 4. We have 4 band files here, and this for loop will loop those 4 band files and convert them into 4 BigWig files. Okay. Okay, so we will generate those BigWig files. I already did that, so it takes some time. So I'm not showing here. I'm not going to demonstrate like in real time, but in the end, you get this 4 BigWig files. And with those 4 BigWig files, you can actually load them to IGV and look at the signal, actually. So let's see whether I can. Oh, I think I already have it here. So if you open IGV, then you say file, and then load from file, and then select those BigWig files, and then those are the BigWig files. Okay. And you can see, like just one example here, in the CCN2 region, you have actually all YAP1 TADs and TAD4 binding at the promoter region of this gene. So you can zoom in, zoom out, and you can see okay, the binding pattern in the genome for the transcription factors. Okay, and as I said, like this is just, if you remember, in BigWig files, it's just, okay, you predefine the bin size and just count how many reads in that bin. So if you actually zoom in, you will see those are just histograms, right? And each bin here is actually the bin size. So I believe it is 50 base pair bin size. That's actually the default. So if you just this little thing here, this little thing here, this is 50 base pair, and for each 50 base pair you have a constant number. Okay, so there's no actual mystery. This is just a histogram. Okay, the BigWig files. Okay, so once you have the sorted band files, now you can call peaks. So peaks, so we have the raw signals actually in BigWig file. You know, okay, you can look at them in the genome. Where are they? But you want to identify all these regions like in the genome actually computationally. So that's actually what MAX2 or MAX3 is doing. So I think this most recent version is MAX3, but MAX2 was really widely used, highly cited tools for identifying enriched signal or ATAC-seq signal, for example. Okay, you use pip install MAX3, and after you install it, then now you can call peaks. So that command is MAX3 call peaks, the subcommand. The treatment will be the transcription factors, the band files, and the control will be the IgG control file, sorted band files. And we tell them the format is a band file, tell the name of the folder, actually the prefix of the file, and then the genome is human, Homo sapiens, and then out directory is YAP-Pig. For example, you can give a different name here. So after you finish this, then you have this actually 5 folders, and then you have the prefix output here, the bat files and the nano-pig, and there's also an Excel file here. So you can take a look. So I have done this. So if you look at the YAP-Pig here, now you have all those files here. And then if you look at the dev files, actually nano-pig file, so it tells you, okay, this is chromosome 1 here to here, this is the YAP1 pig. And there's some other actually statistics calculated from MAX3. There's some p-values and q-values here, okay? And again, this file, you can directly actually upload it into IGV and then you can visualize it. So that's what I did here. So then you see here, those are the peaks. Okay, YAP1 peaks, for example, this TAS peak. Those are the peaks identified by MAX3. And so what we can do here again, so if we want to call peaks for all these 3 transcription factors, now you can use the same trick, the for loop. But now it's a little bit, there's a trick here because we always call peaks against the IgG control. So we loop all the sorted band files, but only if the band file is not that IgG sorted band file, right? And then we essentially call the MAX3 commands. And there's some actually batch stream manipulation here. So for example, like remove the sorted band files. So for example, like here, the sorted band, remove it. So it becomes, the name will be just tags. And then here, the outer, okay, you replace the dot sorted band file to peak. So it will become this. Okay, those are just bash tricks here. So essentially, if we do this, so you see the echo commands here, so it will only print out the commands rather than run it. So if we do here, so essentially those are the 3 MAX3 peak calling commands that we're going to run. Okay, so call peaks, this is the control, this is the treatment, this is control, the format, this is the name, this is human genome, output directory. Okay, you can name them differently, but that's how I name them. And if you remove that echo and rerun this commands, it will actually create the PIC files. Okay, so yeah, that's how I did it. So what we can do, okay, we can roughly check, okay, after you call PICs, you usually want to see how many PICs are there in the whole genome. So you can use this actually find command. It's a very powerful command. So if you use find current directory -name and then wildcard PIC, so it will find all the files that ends with PIC, okay, and it will search all the files recursively inside the folder. Okay, so if you do this, okay, to find actually this PIC files within this folder and this PIC in this file. And what you can do, you can pipe it to xargs, and then you can see what count, and it will tell you, okay, for each file how many lines there. So roughly you get 10,000 PICs for each of them, which is very reasonable for those transcript factors. Okay, of course, it's always good to just visualize them on IGV and to see how reliable those PICs are called. So for example, you see, okay, those are pretty reasonable, right? So across the genome, so you can take a look across the genome for multiple regions here, for example. Okay, I don't know, like for example, you see those are the PICs called. Those PICs are called here. Like we can zoom in here, and you see, okay, indeed, okay, those looks like real signals, right? And the last here, this is actually IgG, which is the control. You should not see a signal there. So if a PIC is called, okay. Okay. Yes, there are some more details about the format of the PIC file, the nano PIC, kind of explain what are the columns in that file. Okay, the p values and q values, all those stuff, and also the relative summit position to the PIC start. So where is the highest PIC signal? So if you go back to here, and you see here, this is the highest signal, like the summit, essentially. Okay. We can look at another example here. So let's look at here. So for example, this is the highest here, and this is the highest like signal here. Okay. Okay, those are called the summits. Okay, there's no actually mystery about that. Okay, and I think that's it for today. I think starting from my next video, I'm going to show you how to reproduce all the figures, and that's the fun part, how to actually use all the files that we already had created by those Unix commands and create those beautiful figures in the paper. So if you go here about section here, like we're going to create or recreate all those figures, within R. Okay, that's it for today. I hope you find it helpful, and make sure you subscribe, and I will see you next time.